Welcome to Overheard of Frequency, a podcast exploring issues surrounding hearing loss. I'm your host, Colleen Chapman, and my goal is to bring timely and interesting stories about hearing science, advocacy, and innovation to our listeners. Frequency Therapeutics' mission is to develop therapeutics to regenerate hearing that may one day improve quality of life for individuals with hearing loss. There are exciting new things happening in the hearing health space beyond hearing restoration efforts, and we'll cover some of those compelling stories too. This is the second of a two-part episode in which I sit down with Carl LeBel, Chief Development Officer at Frequency. Carl and I discuss new information that was shared during our first ever research and development event on November 9th. We talk about what we are seeing in our massive database of trial data and how we are using that knowledge to understand the ideologies and severities of hearing loss where FX322 may have an impact. We also discuss a new therapeutic candidate for hearing loss, FX345, which has provided some interesting results for us preclinically. And we explore a new piece of our clinical trial work focused on patient-reported outcomes. Hi, Carl.
Hi, Colleen. Good to be with you again.
Yeah, good to be with you too. So we promised people after the last time we were together that you and I would get together again for a second episode, which we would do right after our R&D day so that we could really brief the patients and other listeners on everything that we shared during R&D day, which was actually quite a rich mix of both what we're seeing in our data and what we're doing going forward, and not just in the hearing space, but also how we're taking our science into the space of multiple sclerosis too. So kind of exciting things there. Let's jump right in because there's actually quite a lot that I wanted to ask you about and some questions that are emerging in social media from patients too that I wanted to cover. So we shared, as I said, some information on the advances in our hearing programs and the MS work that we're doing, but you covered a lot. You were a big piece of that day. And what were you most excited to talk about, most excited to share that we hadn't been able to share before?
Well, it was amazing to be able to see it all come down into one program. I know that it represents, years of work, but in my mind, it's really three things that I think that mattered most to me. Firstly, all of the work that's gone into preparing for the 208 study, the new phase 2B study that is now underway. It took some not so positive results to help inform us how to best design that study. And then all the data that Kevin presented with pooling, all those analyses and the assumptions that we used to carry forward, all that really culminates in the 208 study. So we're pleased to now be making progress on it. I think the second thing is the announcement that we made about advancing FX345. So that's a new molecular entity. It's a new drug combination, but we think it gets better distribution into the cochlea. And so we're hopeful that might allow us to access other groups of patients that we might have limitations with on FX322. And then the last thing, as you said, the MS program is just really exciting to be able to leverage the platform that we've been using where we've been studying progenitor cells. And in this case, we're looking at progenitor cells in the brain that are responsible for making myelin. And so to see each of those programs advance in a relatively short period of time is, I think, just an amazing thing to be able to experience with the team.
Yeah, so you mentioned a few things when you covered all that, Karl. One was the pooling of our data, which I want to get into much more detail because it shows some things I think people may be very interested in terms of the collating of the data and what we're seeing in terms of patterns. I also want to give some time later on to talking about 345 because the patient community since R&D Day has had a lot of conversation online on social media about exactly what this new candidate 345 means for us and for them. But let me talk first about what you shared from our other trials. We were very transparent. You were very transparent about some of the things from the previous trials. And based on that, what does give you confidence that the hearing signal we are seeing, we feel we are witnessing is real?
Important questions. And so it actually goes before the clinical trials. That's sort of what gives me confidence. When I first was aware of the program and I reviewed all the data that was part of the IND for FX322, I saw data, in vitro data, which showed that you could put FX322 into cell cultures and you could show this nice proliferation of the progenitor cells. And that's sort of the first step that was proof of concept in vitro, though. And then I shared in my presentation some in vivo data in a preclinical model where we did a noise damage model. You could see a restoration, firstly, the damage to the sensory hair cells, and then following FX322 administration, you could see a restoration of those cells and a return of function. And that work is in press in one of the journals that I cited. So
yeah, I love that image. Yeah.
That really powerful image, and but you can't see that when you're doing clinical trials. So the next step then was to get to the trials, and I think what gives us the confidence is we've now seen this signal in 2 separate studies in the same patient populations. So that's pretty impressive, and I think gives us greater confidence now as we go forward with all of the new study design issues that we shared yesterday. That I think that we've got a high likelihood of success here as we keep advancing the program through development.
Yeah, and there's some visuals you just mentioned one of them, but there are a lot of visuals that we shared during the presentation that we'll share throughout the podcast because I think they help people access what we're saying even more because it gives a form to some of the concepts that we've been talking about in a much more defined way. One of the main things of R&D Day, Carl, is that we shared an enormous amount of data, more than we could possibly cover in this podcast, and we'll share a link to where everyone can find the slides to contain all of that data that we walk through. But that's an illustration that links to the fact that we have the largest database of any hearing therapeutic candidate from trials. And one of the things that comes from that is really starting to go through the ideologies and the severities and to start to hone in. So can you talk a little bit about that and the ideologies and the severities that we have been evaluating?
Yeah, in a couple slides, we showed a grid, and that grid is what we've used. And as I said, in order for us to attempt to get a broad label or broad indication for FX322, in our conversations with FDA, it was clear that we needed to work our way around that grid. And that meant studying all of the different severities, which go from mild, for us, it's mild to severe. And then the etiologies, there's really 3 main etiologies. There's more in addition to that, but the 3 main ones are noise-induced hearing loss, sudden sensory neural hearing loss, and then age-related hearing loss. There's also folks that have lost their hearing by being exposed to chemical agents, pharmaceuticals. But the big 3 categories are the first ones that I mentioned. And so we have done studies in all of those etiologies, and we've done studies with patients across all of those severities. And that using that grid, we've been able to really hone in on the patients that seem to respond the most. And there seems to be a pattern of a couple of etiologies, specifically the noise-induced group and the sudden sensory neural hearing loss group. Those are the 2 that have shown response. In the age-related study, we did see 1 patient respond. So we're not giving up in that category. We just need to study it further and understand it a little better. Then as we swap to the severity side, most of the activity occurs in the moderate to moderately severe range. But we've also seen patients, and I described it as high mild category and then the low severe category.
Yeah, and I think it was so fruitful for the field that the FDA asked us to cover all of that space, all of the severities, the etiologies, to really examine all of them. And now you're right, the circle really does show where we're focusing. But one of the questions that's also rising is, and you touched upon it a little bit, for those who aren't in that focus area right now for 208, what might the future hold for our work in those other areas?
Yeah, we've been doing clinical trials with FX322 for maybe 4 years, roughly. And we're still in the
I
beginning.
know, it's amazing. It's
Right?
a short period of time. Really is.
Short period of time, and you've seen how much data. We presented all of the
Yeah.
data yesterday. So there's a lot of data, a lot of learning still happening. So when you develop drugs, the key is, and we talked about this previously, you got to really understand what are the patients, where is the signal being observed in what patients, what are the characteristics of those patients. So while we're focusing first in these etiologies, that's where we want to keep driving forward to try to get the drug approved. But we're going to continue to expand beyond that. So this is the way that drug development programs are run, and we're kind of following that process. So, we're encouraged the way that we've seen response to the drug in those patient groups, but we have an interest in continuing to expand beyond.
Yeah, well, that's where your 30 years in drug development becomes a real asset too, in terms of leading through all of these phases. So now that we talked about the pooling and the data, let's talk about the measurement a little bit, because sometimes it's hard for people to contextualize what the measurement means, particularly those who don't have hearing loss. But even with hearing loss, this 20/20 vision is one thing that people really understand, but the measurements around hearing and hearing health and what different movements up and down that scale mean for people in their daily life is much harder to contextualize. So can you walk through a little bit in terms of some of what clinically meaningful means in terms of potential changes to people's daily lives or changes to what they may use to assist with hearing in their daily lives?
Sure. When we started our clinical trials and we were talking to individuals that had hearing loss and just trying to understand how they described their own condition, the way that hearing loss is described in the field, in the audiology field, people use things like pure tone averages, and it's an average across a number of frequencies, and it's hard for patients to understand what that means. And Kevin Frank spent a lot of time introducing the 10% concept. So let's just take some examples, and I think Kevin spoke to some of these. But if you walk into the clinic and you do the speech perception test, so here you're getting words presented to you, 50 words is usually what we use, and we're recording the number of words that you're getting correct. So if you start the study at, let's say, 60%, you've gotten 60% of 50 words correct, or 30 words out of 50 you've got. That's where you start. Now, what the field is used to is all that happens is people get worse over time. Your hearing just gets worse over time because there's nothing to help today. So if you go from, let's say, 60% and you lose 10% and now you are at 50%, that means you're going to have trouble communicating in a noisy background. There's no question about it. And being able to use a telephone is going to be challenging. That's what people have been experiencing. Now what we see is the opportunity that with FX322, you can improve by 10%. So what does that mean for that person that started at 30 words correct? Well, they got 5 more words correct. So now they get 35 out of 50. And now they can start to hear better, in particular in noisy backgrounds, and their communication is just going to be better. So that makes a difference in their lives. So if we look at the other side, if somebody is in the 30 to 40%, let's say as a starting point, if they go down by 10%, if they've lost 10% of their function or 5 fewer words, they only have one option left, and that's probably a cochlear implant. But if they can increase by 10% and get 5 more words correct, then now they can use a hearing aid to assist them. So you can see that one of the opportunities for FX322 is potentially to fit in with the use of devices as well. And we continue to be open to that. But the last thing I want to say about these percents and such, and as I said, Kevin spoke about this somewhat, we have people in our trial that are showing 20 to 40% improvements. This is sometimes a doubling in your word score, getting twice as many words correct than when you started the study. That just has to go a long way to help people, and that's just the work ahead of us. But we're really excited that the improvements are in the very thing that means the most to patients, and that is better speech perception. They can communicate better. And we're just thankful that that's where our drug acts.
So let's talk about then I was going to ask this a little further along, but since we're talking about patients and their quality of life, let's talk also about the patient-reported outcomes, which we haven't covered on any other podcast to date about exactly what we've created to capture the patient perspective from their trials and from their life. So can you walk people through what we've created for patient-reported outcomes and how we're engaging patients in the trials in that specific way?
Yeah, we've been working with patients for the last couple of years now on this patient-reported outcome measure. It's a questionnaire, and we call it RADIAL, RADIAL. It's an acronym. And the requirements today for drug development, FDA has guidance documents around this, and they say that if you're going to be capturing those subjective measures that patients say how their condition alters their daily activities, and can you determine whether a drug can improve their daily activities. To use measures like that, they absolutely have to be developed by patients. And so we've been working with experts in the field who have done this in a number of other disease areas. And now for the first time, we're developing Radial. It's still under development. It's not a final questionnaire yet, but we've been working with them, and there have been hundreds and hundreds of patient interviews that have taken place where people describe what are the things that affect them the most. How does hearing loss affect them in their lives? Does it affect their relationships? How does it affect communication? How does it affect the ability to understand the clarity of sound and the details of sound? Are there safety issues that they're concerned by? So all of this information has made its way into Radial. Right at this point, it's a 47 questionnaire, and we're still working through it. It's now going into our trials. It's in our 208 trial, and we're going to be using that trial to validate the instrument. And the hope is, in the end, what we want to be able to say is, when you give FX322, you can show some improvements in speech perception. Now, how do those improvements in our clinical endpoint, our primary endpoint, how do those translate into somebody's life, right? Into how their days go? And that's the hope, is that patients will be able to report to us in their own words how their lives have been changed by the potential improvements that maybe FX322 can provide.
There are a lot of questions in the patient community about how does 345 relate to FX322? What's the timing of things? They're eager to hear more because it's a new piece that they're just learning about. So can you say any more about 345, or can you talk any more about the relationship between 322 and 345? We'll interview the team working on that on a future podcast, but even a little bit or a glimpse you can share would be helpful to people.
Yeah, I can share a couple of key things, and what a fantastic team that's been working on that for the better part of a year at least now to get it to this stage. So what we've done is we've swapped one of the molecules in the combination. Now, by swapping, what this different formulation, FX345, does is, because of the chemical characteristics of the two drugs together, it gives us much more flexibility in the formulation. And that has enabled us, at least in preclinical studies, to now see that we can get FX345 distributed at in a much greater area, or further into the lower frequency range than we can with FX322. And part of this is just the combination of putting two drugs together and the characteristics of what that formulation does.
Great. And we look forward to talking to that team in a little bit. As I said, we'll interview them in a few months on the podcast. Let me talk about, since we're talking about the way that the drug works, there has been some confusion in conversations that I've had with various people over the last few months about regenerative medicine and gene therapy. So can you share a little bit about what the regenerative piece of our approach actually does and how it's different from gene therapy.
Yeah, sure. So the approach that we take, and I talked a little bit about this in my section, what the hearing biology team discovered is that these two drugs in combination target what we call progenitor cells in the cochlea. And those cells they're pre-programmed stem cells. So their job is to do a couple of things, and one of the most important things is in development, and particularly when we're in the third trimester, that's when our sensory hair cells in our ears form. And those progenitor cells are active during that phase. They form cells, and then we're born and we have hearing function. But what's interesting is those cells turn off once we're born. They don't keep regenerating like other tissues do. That's something unique about mammalian inner ears. They just don't regenerate. So by providing these two drugs that make up FX322 and soon FX345, those drugs activate the cells and signal to the cells that they're in development still. And if they think they're in development, what they do is they make new sensory hair cells. And it seems if that is the mechanism, that's what our biology has told us, then that is being manifest as improvements in speech perception, which is just a wonderful scientific and medical story to be able to talk about, right? Now that's very different than a gene therapy approach or CRISPR or delivering a construct of a gene in a vector of some kind. You're not, in our case, we're not altering the genome. We're not altering it in any way. But that does happen if you use gene therapy. You are changing the genome. But the approach that we take is unique.
And I think that I love talking about the way our therapy works, because I think when you do talk about the fact that we're born with those hair cells and they're damaged over time, and evolution wasn't prepared for the noises that we're exposed to since the Industrial Revolution, then people really, really begin to visualize it. And then to say that there are dormant cells that we're activating, the science becomes really cool to them. So gene therapy is also really compelling science. But I think it's helpful for people to distinguish between the two. And also, when we talk with Sue about regulatory at a future date, we can talk about the regulatory process too that regenerative medicine has. So, well, that's everything I wanted to cover. It was a lot. It was a lot, Karl. So thank you. And it was a lot on the heels of addressing it, a lot of it during R&D Day. But I'm glad we had a chance to talk about it in a different way, because within the context of patients, it's a very different context.
I could talk about this all day. It's just so exciting to be part of this program and to have the hope that we want to bring to patients that have acquired hearing loss. That would be just a remarkable feat, and we feel very fortunate to be part of that.
Yeah, and anytime we have a trial that's actively enrolling, it's really exciting too. So we will add on to the end of this podcast again how patients can find the information to speak with the screeners and see if they qualify for 208. It's just a really exciting moment for us, and hopefully for the patients as well. So, well, thanks, Karl. Really appreciated it.
I enjoyed it.
All right.
Okay.
Talk to you soon.
Bye.
We hope you enjoyed this episode of Overheard at Frequency. Many thanks again to Carl LeBel for joining us. Please be sure to subscribe to this podcast on YouTube or wherever you listen to podcasts to hear more conversations like this one. Overheard at Frequency was created by Suzanne Day, Colleen Chapman, Jason Glashow, Michael Schubert, Dina Krug, and Zach Russo, with production, editorial, and design assistance by Greg Chen, Victor Burbison, and Zuri Lowell. Thank you for listening.